Hello class, my name is Josh Dougherty and today I will be teaching you about the ion channels and neuronal communication. So in this presentation, I'm going to be teaching you folks about the ion channels, and we're going to be exploring the different properties of these ion channels. Also, we'll be summarizing the voltage-gated ion channels, which we'll be focused on since it will have an effect on the electro communication. And we'll talk more about that later. And then we'll be discussing about the neuronal communication in greater detail. And I will go over and explain how these gated ions will underline the signaling. And finally, I will show you how ion channels generate communication. So the main function for these ion channels is that they allow only inorganic ions. And these ion channels will catalyze rapid transport for many of the salt ions I've discussed, such as sodium, potassium, and even calcium. And there's even one mentioned of chlorine. And their main function of these ion channels is that they will allow these ions to come across the cell membrane here, and they will allow the ions to go within the cell itself. So basically, they allow these ions to go across the cell membranes in order to enable electrical and chemical activity in the body. And a little fun fact here is that one of the cool benefits of ion channels is transport efficiency. In fact, these channels have an advantage over carriers in that it will allow up to 100 million of these ions. I mean, can you imagine like a lot of cars just going through this long highway? They allow millions and millions of them just pass by easily. Well, think of it that way. And they can pass through this one open channel each second. That's a lot. And so they allow all these ions to just pass through easily. Okay, so while the ion channels do have some cool benefits, one thing that they can't do is they couple energy sources. So they can't really do that. And this coupling of energy sources is important to perform active transport. So the transport that the ion channels mediate is always passive. It's downhill. Thus, the function of the ion channels is to allow specific inorganic ions, as I mentioned earlier. It's not where they need energy to let these ions pass through, and it's a passive. Basically, it's a simple toll-free bridge or tunnel. Actually, I would say it's more like a toll-free tunnel, and mainly it allows sodium, potassium, calcium, even chlorine, as I mentioned before, and they diffuse rapidly down their electrochemical gradients across the lipid bilayer, which is very important in order that the cells will be able to have all these ions to pass through in and out, and if necessary. So what I want to talk about now in this slide is these ion channels have two distinctible properties that make them different compared to aqueous pores. The first one in which I want to talk about is what's known as ion selectivity. Now, ion selectivity is they permit some inorganic ions to pass, but not others. And they're pretty much selective. Okay, hey, you can go, you can't go. So they're pretty particular, which is a good thing because you just don't want let all these ions to just come in at once, and it would just be disastrous for the cell to function, let alone neuronal communication. It just won't work that way. It's like electrical circuits to where you let one passage of electricity to pass through and the other to go in that direction, and too much of it can be a bad thing. and you don't want that to happen. So what this important is, these pores must be narrow, okay? Narrow enough in places to force permeating ions into intimate contact with the walls of the channel, all right? And so that only ions of appropriate size and charge can pass. In fact, this is done by shedding the water molecules. So up here we have this little ion here, okay? And it's carrying these water molecules, nice and attached. But this little narrow passage, it ain't gonna let this molecule pass. I mean, it's gonna let go of the luggage, okay? Imagine yourself carrying all this luggage. It's like, you're passing through this narrow passage, like, I can't get through that. So, boom, you drop in, you go through that passage, narrow passage, but you get them back eventually. You just need to let them go so that you get the water so that you can get through, okay? And up down there is like the ion passes through, and it actually gets its water molecules back, in which the gate will open. You just get through the cell membrane and inside the cell, and that's pretty much how this works. Okay, so like I said earlier, the permeating ions have to shed most of all of the water molecules in which they're carrying in order to pass through these narrow parts of the channels. And these ions have to go through a single file. And this process is called selectivity filter, which is a limiting way of cutting down the rate of the number of ions passing through the cell membrane. And while this is happening, as the ion concentrations increase, as there's more passing inside the cell, the flux of ions through a channel increases. So the more ions passing through, the greater the flux. But eventually that levels off. It saturates at a maximum rate. Like once it reaches to a certain level, like to a maximum level of ions, it will eventually just stop, like a maximum rate. There's a certain limiting point here. And one thing to keep in mind is you don't want to have too many ions to pass through. And that is to make sure that you don't have too many ions, because otherwise malfunctions could occur in the cell and the cell membranes will not function normally. And so, okay, so the second important distinction between these ion channels and simple aqueous pores is that the ion channels are not continuously open, and I'll talk a little more about this, and instead they are gated, which allows them to open briefly and then close again. All right, so over here we have these voltage-gated ion channels, in which I'll be talking a little more in detail. And so in these voltage-gated ion channels, a change in the transmembrane potential causes a charged protein domain and to move relative to the membrane, opening or closing the channel. And by the way, both types of these gates can go very fast. And so over here we can check out this image. Well, during the resting or the closed, you see here that the outside of the cell membrane is positive. That's the extracellular side, and then within the cell is negative. Now, once the gate opens up, all right, the ion here, like for example sodium, will actually go within the cell, thus making it positive, all right? And thus, the less sodium on the outside, the more negative the other side of the membrane becomes. And this gate's a little slow, and once the ions are in, it'll slowly close the gate here, and thus the ion channel would become inactivated. And of course, it rinses and repeats itself through this cycle here. So these voltage-gated channel, the calcium channels, they may function. here's a little fun fact about calcium channels function. They can function at both pre- and post-synaptic terminals to allow calcium influx that either augments neurotransmitter release at the presynaptic terminal or contributes to postsynaptic neuron depolarization. And that plays a role in electrical communication, actually. And this is one of the reasons why calcium is very important to have in our bodies. It's not just to build big, strong, thick bones, but also it's good for neural communication. Because, well, otherwise, how can electrical energy pass through us? So, with that in mind, however, too much calcium is a bad thing, as excess amounts of it could cause some bodily malfunctions. Like, for example, excessive calcium influx at postsynaptic termini might impair long-term potentiation. And large conductance calcium-activated potassium channels at postsynaptic terminals and small conductance calcium-activated potassium channels at postsynaptic terminals quench membrane depolarization by allowing potassium efflux, which facilitates neuron repolarization. So, interesting facts about these things. So, obviously, we need the nerve cells to have neuronal communication. After all, it's part of the nervous system, and the whole part of neuronal communication is to have nerve pulse signals to contact from the sensory or motor neurons to the brain. And so, with that said, the nerve cells need to have ion channels because they need to use these ion channels for such depolarization, repolarization occurrence to occur. And they have made a specialty of using ion channels, and I'm going to consider how they use a diversity of such channels for receiving, conducting, and transmitting signals. And certain cells in multicellular organisms are excitable. I mean, they're ready to send a direct signal. It's like, ooh, ooh, send me a signal, pooh, pooh, pooh. And they pass it on. And they play a great role, a major role in nerve conduction, because without these exciting neurons, well, can you imagine having a dull neuron cell to where it's like, I don't really want to send this signal. Meh. And your body wouldn't function. And I think that's one of the reasons why sometimes when your body feels numb, like your foot to sleep, or actually one case example of what happens when your body becomes numb is your nerve cells get into that state where they don't send any electrical signals. Like, for example, you been into the dentist to where they eject a shot in your gums to make it numb? What that's doing is it's preventing that neural communication from occurring so that you don't feel any pain. Usually it's like you stick something in, ow, ow, ow. You will feel it, feel it. But with that shot, the numbing, it's like, well, you don't feel anything because of numbing them nerve cells. So nerve conduction cannot occur because of that. So that's just a little fun fact there. Okay, so with that said, we're going to be talking about how gated ion channels underline the electrical signals here. And this is a simple little process of how, in this picture, for example, you have sodium ions out and the potassium ions in. And this is all about the sodium-potassium APT pace, which, by the way, is electrogenic. What that means is it creates this charge imbalance across the plasma membrane, okay, by carrying the sodium out of the cell, as you can see, for every 2 potassium ions coming in. So the action of the ATP pace makes the inside of the cell negative relative to the outside. As I showed you earlier, remember how the ions come in and it changes the charge of the membrane. Well, here, this makes the inside of the cell negative, and then this more positive for that process. So inside the cell, potassium is higher and sodium is much lower than the outside of the cell. And the direction of spontaneous ion flow across a polarized membrane is dictated by the electrical chemical potential of that ion across the membrane. And I'll talk a little more about that in the next slide. So with that said, this has two components of this electrical potential. Okay, so the first potential is the difference in the ion concentration. I mean, there is a difference on the two sides of the membrane, and the difference in the electrical potential, typically expressed in millivolts here. So given the ion concentration differences and the millivolts of about negative 60 millivolts, all right? So which is inside negative. Opening of a sodium or calcium channel will result in a spontaneous inward flow of sodium or calcium, which causes depolarization. Whereas opening up the potassium channel will result in spontaneous outward flow of potassium, and which causes hyperpolarization. So such effects are important for the electrical signal to flow. So in this case, potassium moves out of the cell against the electrical gradient because the large concentration difference exerts a stronger effect than the millivolts, the action potential, the electrical potential, that is. When it comes to chlorine, now chlorine's interesting. Kind of a bit of a minor role. So the membrane potential predominates, all right? So when a chlorine channel opens, chlorine flows outward. All right, so when it comes to the electrical signaling of these nerve cells, this is accomplished by the important neurons. And these neurons play a special role to carry electrical impulses. They are the electrical signal carriers. They pass it on. And these neurons make sure that these electrical signals go all the way from, let's just say I touch my finger here, to it gets to the brain, to where I feel it. I feel something. Look, Josh has touched his finger. So it's this messenger, basically. It sends out messages, and they carry an electrical impulse, which is the actual action potential, and they send these out from one end of the cell, which is cell body, through an elongated cytoplasmic extension. That's an axon. Think a wire. Like, think of the little neurons have these electrical energy cell that sends these signals in a long wire to the other energy cells. Think of it that way. And so the electrical signal triggers release of neurotransmitter molecules at the synapse, carrying the signal to the next cell in the current. So with that said, there are three types of ion channels that play a role in such neuronal communication. I mean, you have the sodium ion channels, which allow sodiums in the cell. As you can see here, the sodium enters through the axon, which causes depolarization. And then you have the potassium channels and the calcium channels. And they play a role in such communication as well. So sodium channels cover the entire axon. And we see here, they have these channels on the node of Revnier. And they pass through the myelin sheath, which, as you noticed, they don't have any of these channels. They don't have them. And it's not necessary to have a bunch of these ion channels throughout the axon. They just need a certain amount because the sheath plays a role of conducting the electrical signal through the axon. And of course, the channels are closed when the membrane is at rest, which is important to ensure that just the right amount of sodium enters through the cell. But of course, these sodium channels do open briefly when the membrane becomes depolarized locally in response to acetylcholine or some other neurotransmitter. Also distributed along the axon are the voltage-gated potassium channels, and you can see those here. In fact, they actually are underneath the myelin sheath like we looked at earlier. You see at the node of revenue here with the sodium channels, and then you have the potassium channels in the myelin sheath. So with those channels, they open a split second later in response to the depolarization that results when nearby sodium channels open. And the depolarization flow of sodium into the axon influx is thus rapidly countered by a repolaring outward flow of potassium, the efflux. That's what it's called. So at the distal end of the axon, which we're going to see, are voltage-gated calcium channels. We'll see that soon. And these channels, of course, when the wave of depolarization occurs and repolarization happens, is caused by the activity of the sodium and potassium channels arrives, and triggering release of the neurotransmitter acetylcholine, which carries the signal to another neuron. So it's like firing action potential, or to a muscle fiber. It's like contact. So whenever the sodium potassium channels do their work, the calcium will release these neurotransmitters to cause the action potential to go to the next, like fire at will. And so that's how it actually. All right, so continuing with the same image here, we're looking more into the voltage-gated sodium channels, which they are selective for the sodium over other cations by a factor of 100 or more. And they also have a very high flux rate of over 107 ions per second. This is per second, mind you. That's a lot. And a sodium channel that opens in response to a reduction transmembrane electrical potential, it closes within milliseconds. That's very fast. They close very fast, and remains unable to reopen for many milliseconds. And as I said earlier, you don't want too many ions to flow through, so to prevent this from happening, they had to be very quick. Open quick, because they're letting a lot of ions, and you don't want too much sodium. And this is one of the reasons why too much sodium in the body can be a bad thing, especially for the cardiac nerve cells that are connected to the cardiac muscles, because yeah, that could cause some problems here. So this is why it's important to make sure that these sodium ions channels work the way they should, like open, close. So make sure you don't have too much sodium in your body. Okay, so real quick here, we're going to wrap this up. So the influx of sodium through the open sodium channels depolarizes the membrane locally. This causes voltage-gated potassium channels to open up. So the resulting potassium efflux repolarizes the membrane locally, reestablishing the inside negative membrane potential. And a brief pause of depolarization thus traverses the axons, as local depolarization triggers a brief opening of neighboring sodium channels, then potassium channels. The short period that follows the opening of each sodium channel, during which it can't open again. So, like I said, millisecond close, open, close, ensures that a unidirectional wave of depolarization, the action potential, sweeps from the nerve cell body toward the end of the axon. So it's a one-way direction. It makes sure that it goes one way. That is all. It's just final destination should be the central nervous system, which is the brain. Okay, as the wave of depolarization occurs to the very end of the axon here, we have the presynaptic neuron here. The depolarization will cause the calcium channels to open, which eventually will allow calcium to enter, and then you have the acetylcholine to be released in this empty space, the synapse, which goes to the postsynaptic. It binds to the acetylcholine receptors here, which ultimately triggers the depolarization of the next neuron cell. And so that is how neuronal communication occurs, and thus is how electrical signaling by ion channels is done. So thank you for watching this presentation. If you have any questions, just let me know. Have a good day.